अब आपको डॉक्टर गांगुली का वैक्सीन के बारे में जो है कोविड वैक्सीन के बारे में क्या प्रोग्रेस है इंडिया में एंड वर्ल्ड वाइड
What
वो
is
सुनने
new in your nursing home?
nothing is new. I have started admitting patients and I am taking care of that and if anybody is having fever or any long history of body aches, sneezing, headache, we are not admitting such patients. ठीक है, we are asking them to get their test, PCR test done. Otherwise,
Guidelines change हो गई हैं?
regularly patients are being examined in OPD also, and we are admitting also the patients also. With
कितने पेशेंट एडमिटेड हैं आप?
अब मेरे पास 5 पेशेंट एडमिट.
टोटल स्ट्रेंथ कितनी है?
15.
50 की.
15 15
15 बजे का.
50 नहीं है. हाँ, 5 पेशेंट एडमिट हैं अभी.
तो अरुण जी, आप थोड़ा अपने बारे में बताइए. अरुण जामकर जी, क्या कर रहे हैं आजकल?
Actually, after I retired, I joined a couple of organizations. I'm working with persistent systems, where I'm working with as a healthcare consultant.
तो उसमें क्या क्या हो रहा है?
We're actively involved in creating a software for epitome analysis and giving latest versions for say any viral vaccine. And we are presenting ourselves to Serum Institute of India, who have undergone into MOU with Oxford vaccine. So our aim is to get latest what you call as analysis of all epitome of spike proteins, so that when the vaccine is introduced, it should be able to protect Indian population.
So when do you think the vaccine will come?
Yeah.
So when do you think the vaccine will come?
Now, once the permission is available to Oxford, the serum can directly manufacture because they have MOU with them, and we are trying to help them out. Because what happens? Suppose we have 10 strains of vaccine, including that AA1. We don't know whether that has been included in Oxford, and by that time there might be several mutations. And my hypothesis is if we don't take care of these mutations, even though our population is vaccinated, we can be exposed to second wave. So in order to prevent ourselves from second wave, we need to have multiple versions of vaccine, so that the latest mutations are also included.
Who else is working on spike vaccine?
I think there are 5-6 companies on spike vaccine.
And they all are working on spike.
Everybody is working on spike protein vaccine.
No. Somebody will try live attenuated, somebody will try killed vaccine, somebody will try.
But then their attempt is towards spike protein. I think.
And do you think that
antibody protection,
when it will be available?
We don't know.
So for a phase 2 trial, for a human trial, when it will be available? Is it available today or for a human trial?
No. They have an MOU with Oxford. Now Oxford is, I think, already in phase 2 trial.
They are already in phase 2 trial.
The moment they make it available in Western world.
So how many cases they are doing in phase 2?
I have no idea, because that is very secret.
Very secret.
Yeah.
Good.
And only as a consultant for software, we are trying to find out how what's called as epitome analysis can be done.
So Dr. Ashokan, we are waiting for Dr. Ganguly. We have started the 0 hour. So we were talking, Arun is, he was in bioethics, he was you retired as a Vice Chancellor of Maharashtra Health University. Am I correct?
University of Health Science.
Yes, he has retired as a Vice Chancellor of Maharashtra Health University. He wanted to be there, so I specifically invited him. He will give us the inside view after Dr. Ganguly, what is happening in Serum Institute regarding development of spike vaccine in India. So,
Thank you, Dr. Arun, for joining the IMA webinar
thank
on
you.
vaccines. Yeah, it's a pleasure
Thank
to
you,
have you
thank
with us. And
So I
it's
was,
such a big pleasure that the 0 hours are better than the original content of the meeting normally. That is the special charm of Dr. KK also.
No, KK charm is, I was trying to just correlate with him because I was not going to get link. And I thought what are happening latest in vaccine technology. What's called the reverse
and what
vaccinology.
about mRNA? What about mRNA vaccines?
I have no idea because I'm just trained myself to what is required for persistent systems.
And the using the membrane envelope has no value for making a vaccine. You only need either a spike protein or a mRNA.
I have no idea because I was just going through what is our product and what is our platform.
Okay.
So maybe I can get to somebody in case you want to join because we have a huge department of life sciences.
Has anybody else joined us?
Dr. Ganguly should be joining any moment. He just spoke to him. He says, I'm getting ready.
We have JK with us. We have Ramesh Tata. All of them have been muted.
I invited one of my scientists to join. In case
Okay.
she joins, she might be able to talk with you better.
Dr. KK, how many webinars you conduct in a day, sir?
So I had, today I had 9:31, 11 o'clock second, 3 o'clock third, this is the fourth. And then back to back fifth is the fifth, 5 hours.
Oh my God.
Almost.
How do you manage? It's not the question of, managing time. It's a question of managing knowledge, information and content also, is it not?
See, because I'm in touch with COVID starting from 10th of January.
I know that very well.
And from that, I am in touch with every possible thing. So it's easy for me to correlate all of them together.
Yeah. I know when we went to Myanmar,
yes, we had a discussion
you were still
on that.
there with a laptop at the night.
Yes. And now, say for example, I always read about the future. So today, there are some cases of rat-induced hepatitis E. So today I learned that there was some cases. I've been following rat-induced hepatitis E in Hong Kong, and now a case has been detected in Canada. So I start following it up, whether it is likely to become other countries or not. So rat-induced hepatitis E is something which I start writing about it. So and that's how I started writing about the mysterious pneumonia starting of 10th of January.
Okay.
At least a part of our lepto could be that.
Part of?
Part of the leptospirosis. Lepto.
Yes, absolutely.
How common is leptospirosis in Delhi, sir?
Leptospirosis in Delhi is being missed. So even typhus, even strep typhus in Delhi is being missed. There are cases coming in leptospirosis, but they are missed often. But it is not there so much in Delhi. Even then, it is not there. It is basically has to be there in monsoon regions, in areas where there is ocean. So Bombay and Kerala, wherever there is.
Yeah. We have.
Ocean.
And you have to diagnose it in 24 to 48 hours.
Yes, it is
That's
ocean.
The challenge.
So it is the ocean-related diseases. Wherever there are coconuts, the leptospirosis will come.
Oh my God, coconuts?
The coconuts will only come near the ocean.
What has the coconut got to do with leptospirosis?
Coconuts is a basic formula we say in Hindi when we teach naturopathy to people. And we say that if God wanted you to eat coconut water in Delhi, he would have grown coconuts in Delhi. Connected. He has grown coconuts in coastal areas because treatment of humidity is coconut water. Yes, Dr. Ganguly has joined. Dr. Ashokan, Dr. Ganguly has joined, and I ask you to take over now.
Yes.
Sir, pranam. Dr. Professor Nirmal Kumar Ganguly, the former DG of ICMR. So Dr. Ashokan is our Secretary General of Indian Medical Association. And we are basically, this seminar is being live to all the doctors in the country and through the IMA website. And we will also be sending the link of this seminar again to these doctors again and again and again and again. The objective of today, Dr. Ganguly is an authority, former ICMR, and he is an advisor to the government of India in all the matters. And in all possible vaccines, he has been in touch, and he has been in all the committees about the advisories of vaccine. Sir, most of the medical doctors in India are not aware about the basics of vaccine. What are the basis of vaccine, where we are, will the vaccine come or not? Because every patient asks the IMA member when the vaccine will come, and we only relate and correlate and tell the media reports. So we would like you to address all the IMA members about the vaccine status all over the world and in India. Dr. Ashokan, can you greet him and then he takes over?
Yeah, no, it's a pleasure to have such a senior people with the Indian Medical Association today. We look forward to the discussion, and today we will see that this is put up in any number of channels and an IMA website, so that it is useful for many of our members. Welcome to you, Professor N.K. Ganguly. I also welcome Dr. Arun, former Vice Chancellor of Health University of Maharashtra. Over to you, Dr. KK, for beginning the session.
Sir, I request you to deliver your address, and then we will have an interaction.
Thank you, Dr. Agarwal. Firstly, I congratulate you and I to give right kind of information which are very well analyzed. At this given moment, the problem is that because of the internet knowledge, huge amount of information is coming to newspapers written by the journalists who do copy paste, and many times the insight is not available. I have hardly seen scientists writing an article in an important area, although there are several features on vaccines have come up, but they lack the depth and they lack the incisiveness which will provide some answers. So I will start with one of the questions which you asked, that a vaccine is absolutely necessary for COVID-19 or not. Actually, the main need of vaccine comes under certain situations. Firstly, when there is a large number of asymptomatics moving around the community, who cannot be really treated, because if you start treating asymptomatics, there are huge amount of issues. Secondly, when you do not have enough drugs, and we really don't have at the moment, although remdesivir we have talked a lot, but if you really ask us, we do not have a drug at this moment, although there are a lot of repurposed drugs, but they are acting in certain percentages, and certain percentages are not actually responding to the drug. So in this situation, vaccine becomes the only option. There is another reason for vaccine. We know that the immunity in population is very, very low. Although there are around 1 which has the highest number of cases, the antibodies in population has come up to 25%. This is not enough to stop transmission. If we really do some kind of a modeling which John Hopkins did for HIV/AIDS, if we reach 50%, we will protecting at 70 of this virus. So we need not have 100% protection. This vaccine, whenever given, will also lead to herd immunity in the population.
So your voice is getting disconnected.
And that was the access to vaccine and the acceptability of vaccine in the populations is not always optimal, and people actually resist vaccination. Some of the population
So your voice is getting disconnected.
resist may become their reproductive abilities, etc., will come down. So there is a overall resistance against vaccination, and in such situation we need to create vaccination which will also promote herd immunity. If we wait for herd immunity to develop under the entire population to 60% level or 70% level, it will be about an year's wait or more, because in India, herd immunity will be much less than the other places. So at this given moment, vaccine is important and necessary. So what could vaccine do really? One thing which vaccine can do, that we know that there are the two spike proteins, spike protein 1 and spike protein 2. There are three leafy structures which actually conform and they bind to S2 receptors. Now, around the S2 receptors, there are, for COVID-19, it also binds to some other receptors known as integrin and CD147. So essentially, you block the receptor so that the antibodies to spike protein actually block the spike protein to bind to its receptor, thereby it doesn't enter. The second method of vaccine protection is that boosting of the NK cells, or known as antibody dependent cell cytotoxicity, where the NK cells, they recognize epithelial cells and epithelial cells which are virus infected, thereby again killing the virus. The third method is
उनको बोलो, ऋतु को बोलो,
again
डिस्कनेक्ट कर दे, वो कर दे, म्यूट नहीं, वीडियो ऑफ कर दे।
toxic
अरे ऋतु तो कर सकती है यार।
immunity.
So your voice got unmuted. So your voice got muted. Dr. Ganguly, Ritu, kindly make.
Okay,
Ritu, and also make everybody's video off so that the voice becomes better.
Okay, so
make everybody's video
now
off.
now I have unmuted. My voice was unmuted, but then I have to start again. I
No. Your voice, we are hearing you. Don't worry. It is fine. Go ahead.
We are hearing you. We are hearing you.
So these are the whether the vaccine could protect an individual. So at this given moment, we do not know the exact method of protection which the vaccines will afford. Only one method of protection. All the vaccines which are being designed now are actually aiming to protect it with only one method of protection. That method of protection is through antibodies, which are neutralizing antibodies. Virus may made antibodies will prevent the virus, of spike protein of virus, to bind to ACE2 receptors. So this is a major mechanism at this given moment. We are not looking at other mechanisms. As we get the data from phase 1, phase 2, we will know what are the other mechanisms which are there. So under these circumstances, there are some apprehension that whether the vaccine will succeed or whether the vaccine will not succeed. The two reasons why the vaccine may not succeed, one is this, that this happens in the AIDS virus vaccine development. Till date, we do not have an vaccine against AIDS virus because AIDS virus has a very strong latency, and this latency maintains the virus at the cell, inside the cell, and it is very difficult to kill the virus inside the cell through the antibodies or even through the available cell-mediated immune reactions. So the COVID-19 also has a latency. It hides within the cells and it remains within the cells, as we have seen in the many asymptomatics, where in some of them the virus load is good and it excretes also, and the pre-symptomatics, they also excrete the virus. So the this under this virus which is hiding within the cell will be very difficult to deal with. We don't know whether the current vaccines will do it or they will not do it. The second major problem in this, which we have seen has appeared in the most of the RSV vaccines, and we have also seen that RSV means
यार
respiratory
फोन इतना सारा चीजें ओपन है ना.
Please, please.
so that's it, so that the respiratory syncytial virus vaccine, they promote, they create antibodies which attack the body. The second thing which happens in the like dengue vaccine, some of the antibodies, they attack the immune system, they attack the body itself. In the COVID-19, natural infection actually does two things. It reduces the immune system because there is a lympholysis, and they also have they also act on the phagocytic cells, and it also promotes inflammation through the autoimmune reaction. So this autoimmune reaction is a fear that we do not know whether the antigens or whether the combinations which we are using will also produce autoimmune reaction or not, and will attack our own body after we get vaccinated. So under these three limitations, we are at the moment developing the vaccines. And as we move, we will know whether we are able to this or not, and we will have to create assays in the phase 1 and phase 2, where we will be able to deal. Almost everyone is using their platform through their known platforms. And so nobody is creating a new platform to really deal with it. So there is one kind of a assurance that at least people are using platform with which they have experience. The second thing, what is happening at this moment, which is very, very heartening, that the speed with which we are getting the regulatory approvals or the way the things are moving are very, very fast. And this kind of a lightning speed we have never seen. So there is one fear when we move in this lightning speed, that whether there is enough oversight which has gone into by regulators to move the vaccine smoothly from one phase to other. The third thing which is very important in vaccination, which will be very, very important for us to follow, is this, that the designs are very, very different. Here the first design is 18 to 55 years age group, because this vaccine, this infection gives different responses in the different age groups. So 18 to 55 is the first group in which the phase 1 have been designed. Then there are two more groups which have been designed. One is 56 to 70 years of age, and the third one is 70 years and above. This is one of the most vulnerable group, and there the immune responses are very, very different than the immune responses in young. So At this given moment, the first group has been completed and the results have to be posted. There is a rule that anyone who does a vaccine trial, whether you get a positive response or whether you get a negative response, you will have to put it on your website. So anyone who is following it will have the ethics committees and other experts could go through the data and then take a decision that whether they will be using it or not, or they work with the company as the development occurs. So the second very important thing which is moving now, so the size of the phase 1, each arm, each cohort is 45 subjects. Then the second second phase, which will look at, the first phase looks at the safety. It does some antibody, etc., but it looks at the safety. So second phase is almost 600 people. So there's a very large number of people, and people have thought that they will recruit them with a very fast speed. And in these, the reactogenicity, apart from safety, reactogenicity and immunogenicity will also be measured. Now, as soon as the second, what the companies are doing, which is different from the other vaccine generation, they have started manufacturing the vaccine. Manufacturing plants are being set up. So this is a huge amount of risk taking. This they are doing that if they have a successful phase 2, they will go for an emergency vaccine use. And if we are not able to really control this particular disease, we will go for an emergency vaccine use very speedily and some or other stop it. It has happened in Ebola. In 6 months' time, Ebola vaccine went for an emergency use, although regular use vaccine has yet to come. It is now getting licensed, but it is emergency use was within 6 months. So this is what companies are thinking. But there is a huge risk in this, that if the vaccination fails, then there will be a problem. Then all this will go waste and companies will lose money. Somebody need to protect their investment. Then what will happen that by the fall, actually the fall means around November, December, the emergency use vaccine should be available. And another, in end of 1920s, beginning of 2021, we might have a regular vaccine where the space is from 12 to 18 months. So we cannot predict. So for the best of the effort, if it happens, we will have a vaccine, a regular use vaccine in 12 months or between 12 to 18 months, anytime. And at that time, the vaccine might have gone through all the major important realizations which happens. Now, I will describe some of the vaccines, which are about 120 vaccine efforts are going on. Many of them are startups, and many of them are established companies who are moving it forward. Normally, established companies pick up the startup research and move forward. But here, the majority of them are startups. And there are about 30 companies, 20, 25, 30 companies, which are moving towards the vaccine manufacture. In their assessment, the manufacturing time will be about uh, 8, 9 to 10 months will be the expansion of the manufacturing capacity. It will be expanded in a phase-wise fashion because we will need billions and billions of doses, and it is not possible to produce that kind of doses in a very that kind of expansion in a very short time, because these need to be qualified for GMP, these need to go through WHO pre-qualification, this will have to go through a lot of accreditation before the manufacturing starts, and the product comes out. So it's not a very easy thing, even for an emergency vaccination. There will some amount of certifications will be needed. So this is the time scale on which the vaccines will move. Now, I will like to give you a glimpse that how fast the vaccines have moved. Like the first vaccine sequence came about 10th or 11th January by China. By 14th January, Moderna, the first vaccine which I will deal with is that of a Moderna, because it has completed 18 to 55 years of the first phase. It has completed. It has not put in data. There are some kind of argument which is going on that whether this data, they got everything which they wanted, but the preclinical data was analyzed by Dr. Fauci in his interview to National Geographic, and the preclinical data looked very good. Preclinical data is done through this, that messenger RNA, you choose an area. So here it is known as messenger RNA 1273. So the number denotes that it is an area of the genetic element which will code prefusion part of the spike protein. And then what happens, that this particular thing is interacted for its expression in the cell system. You take convalescent population serum and see that whether it is reacting with them in the population serum or not. Then you go through, there is a specialized mice model which is available in NIAID. You go for a mice model challenge and mice model infection and expression. Then you move to monkeys, and in the monkeys you look at that whether there are ADR reactions happening or not, whether the immunogenicity in monkeys there or not, what kind of dosing is going in there, what kind of antibodies which are being produced. You get lot of data. So Moderna vaccine has gone through all these stages of the development. So the preclinical toxicology was clean, that there was no toxicological issues over there. Based on the dosing and the preclinical toxicology screen, actually 3 different doses have been used here: 25, 150, 250 the microgram of the the doses of messenger RNA 1273 has been used. So now this messenger RNA will be injected into humans, has been injected into humans. Dosing space is 28 days in the dosing space. So this has been injected in the humans, and the reactogenicity in humans, what I know now, that there is a little bit of swelling and there's a little bit of a pain, but it goes on after some times, and the patients do not feel any problem as far as this up to this time whatever has been done. So Moderna, so they have reached this. Influenza and other viruses are tested in the ferret, and all these testing has to be done in the BSL-3 plus or BSL-4 or at least BSL-3 facility, but it cannot be done in the less facility and than other. India at the moment doesn't have any animal models, and to do the primate studies in India will be very, very difficult. So Indian companies who are making it, they are seeking funds and getting it outsourced to test for such things and to access the assays which go with this, as well as the neutralization assay which is done through a host cells. So India, some of the Indian products have moved. So in this situation, as I told you, that within 60 days, the Moderna completed its preparation, the GMP-like preparation, and it actually submitted its product to US FDA for a clearance to go for phase 1. And within 7-8 days, the US FDA gave its permission and approved its protocol for phase 1. So this was the speed. I hope our regulatory bodies and the experts in our country, if I really try to look at experts whom we have it, and if you just ask me the names who will be able to handle it, I'm afraid I will not be able to give you many names on this. So this is one of the things which will be a challenge, and we will have to get it reviewed within our systems very well. Mostly in our federal drug system, many of them are pharmacologists, some of them are toxicologists, but essentially everything has some amount of toxicity, and you have to really look at it and go forward to it. There will be one more challenge in the vaccine development. We know that the women and the men are responding differently in this infection. Children and the adults are responding differently to this particular infection. And sometimes some strange symptoms come in, like it produces a huge amount of vasculitis. So some postmortems are showing this thrombus all around the vessels, the arteries, veins all around, and some people are coming with a stroke. Some people are coming with a gout-like thing. There's a swelling and redness of the thumb and the toes and the fingers. And so colchicine trial is also going on that. Some people some people start with stroke, some people start with a gout-like thing. And one more thing which is coming up, some children are showing Kawasaki-like symptoms. And this is very, very frightening, that when we really move on these, we should be able to handle this as we move forward in our protocols and when we look at it. But at the given moment, the persons who have received injections, up to 2 injections, they have not really shown anything which we should be afraid of. There has not been much reaction. So but we have to move towards the other age groups in the phase 1. It has not happened. This will take some more time, and 600 milligram is the dosage which has been given. So by this time, what Moderna is doing, that it got MORI partnership, and MORI is moving towards recruitment of the volunteers. The volunteers in US are coming up very easily. There is a very good information sheet which is available to that. This will be very, very important in India, so that all the volunteers who come, they are not poor who are coming because they are getting some compensation and some other things. And at this time, the poor have become more poor, and they might join in the trials. So we have to really see that the all stratum of the people with different kinds of nutritional profile, they join in the vaccine effort. So this will be one major area where we need a good public health institute like MORI in our country, with a strong social science group and behavioral science group who should recruit volunteers. It should not be done in a cavalier fashion. So this particular partnership is very, very important, which Moderna has forged. The third thing which has happened, because Moderna is not a very big company, it has to really expand and produce it. So what Moderna is doing, that it has partnered with Lonza to expand its production. So and they have actually started enrolling for the phase 2, and they have put in an application to the NIH for approval of go to phase 2. So there is again something which is very, very peculiar, which is happening. Like the other 2 cohorts of phase 1 is not complete, but they are moving to phase 2. This again hasn't happened in the vaccine world, where it is an NCE. There is again something very, very new. Now let us see who in India is doing this particular effort. There is a company in Pune known as Genova, which is headed by a CEO. His name is, although the Genova is a part of the MCure system. Satish Mehta is the CEO of that, but the CEO of Genova is Dr. Sanjay Singh. He's a product of NIH, and has come back to India to do this. They produce the TNK TPA, which is which has changed the whole value chain in India by providing TNK TPA at a very affordable rate. So they have done their thing is also a messenger RNA system. This messenger RNA system, they are using two things. They are using a carrier which is known as lipid iron oxide, LION. And they are using an adjuvant which they got from IDRI. This adjuvant they got from IDRI, and this is known as glass. And they manufacture the glass now by IDRI's license. And so they have an access to this. This adjuvant has gone to humans. So there will be no issue over there. So they went to US, they have done convalescent sera in the US, they have done the mice experiments in the US, and they have completed the monkey experiments in the US. So they have really moved very, very fast. And let us see that whether we can help them in moving fast and producing this vaccine in a record time. Now, one more thing in the messenger RNA vaccination. Although Moderna has platforms, they have done it in RSV platforms. They have moved it. They have moved it in the influenza viruses, both H7N9 and the other influenza systems. They have used it again pneumoviruses, and they have used into the additional type 2 viruses, influenza viruses. However, none of their system has moved to a vaccine and has been used in the population. So all of them are moving parallelly forward. So this is something which we don't know about messenger RNA vaccination. There is one more issue in the messenger RNA vaccination, that suppose this particular messenger RNA, when it goes, it uses human as a factory. It actually starts expressing the antigen from within the human and produces an immune response within that particular individual. So this is how it works. But suppose we have already pre-existing antibodies in the population and you get the vaccination, this antibody might prevent the expression of the antigens. There is a theoretical possibility. We do not know whether it will happen or it will not happen. And again, we are not using any whole virus and others. So the actually getting ADRs or getting the self-reacting antibodies in this system is little less than those who use attenuated viruses or whole cell viruses in their vaccine design. So this is where we are at the moment with the messenger RNA vaccine. None of the messenger RNA vaccine has gone to human population. So we do not have any experience because it is not being sold in the humans. There is one small niggling problem in this, that messenger RNA vaccines till date has worked in everyone who has a tail, like mice, primate, and everyone. But if you don't have a tail, this has not yet been in use in the population who do not have a tail. So we have to see how it goes. But there's the most promising vaccine candidate at this given moment. And I have given you a parallel in India. The second vaccine which has moved, and which is the other vaccines, I will not go in such a detail as the Moderna one. because there will be not enough time for that, but I will give you some information. If you ask me more information, I will then give you the information. So the second vaccine is the DNA vaccine. Again, DNA, in the DNA vaccine also, the system is the same, that this will start expressing the antigens, analytes into the system and will produce antibodies. They are injected through electroporation or through the guns which are used to move DNA vaccines in humans, subcutaneously or intramuscularly. This vaccine is undergoing trial in South Korea, and IVI, International Vaccine Institute in South Korea, is conducting this particular trial, and they have been funded by the CEPI for this particular trial. Now the messenger RNA vaccine, the first it was funded by CEPI, and the second it was funded by BARDA. BARDA is a US funding organization which funds vaccine. But there is a problem with BARDA. BARDA doesn't allow the vaccine trials at the moment to go outside USA. So this is something, when you get, if you get the BARDA funding, then you will have to deal with this particular thing. The second, so the DNA vaccine, also we do not have a single DNA vaccine in the market. And this is the second promising vaccine. There are two DNA vaccine constructs which are being tried in India by the different Indian companies, and we will have to see how it moves. At this moment, they haven't moved in a speed, so I will not go for it. As soon as the further development occurs, I will inform you. The third vaccine, which is really very, very promising, is through the has been done by the Novavax. Novavax has a platform known as VLP. It is a company in the Rockville. When they were in trouble, the Cadila Group, Mr. Rajiv Modi's group, they purchased a stake in the Novavax, and they created a major company, joint company known as CLP Biologicals in Ahmedabad. So they have used VLP vaccine and put two vaccines into the market. One is the seasonal influenza vaccine, which is a trivalent vaccine. Their quadrivalent vaccine, they got the permission, and they will be manufacturing it. They also have H1N1 vaccine in the market. But what is very important and good is, only yesterday they got permission for the world's first subunit vaccine in rabies, which has also been done in the VLP platform. So Novavax has created this particular vaccine in their virus-like particle platform. Recently, there has also been a very good success for the VLP platform, which has happened in USA, that is for the chikungunya vaccine. And VLP platform, the papilloma vaccines are already in the market by Merck and the GSK all around the globe. So this VLP platform is now have got funding, and it has also been funded by BARDA, but some or other they are able to conduct the trial both in USA and Australia. So Australian trial has moved into phase 1 and moving really fast. If it happens, it will be available in India, because the partnership company which is in Ahmedabad will start producing this. So this has got a lot of value, and we might see a vaccine in India. This will move very fast. Rather, they are just waiting. They are waiting that whether it is successful over there or not. If they get success, the platform is ready. All the things are ready in Ahmedabad. The production will happen very, very quickly. So it's a very smart move by Dr. Rajiv Modi. Dr. Rajiv Modi is a PhD. So he is an entrepreneur and also a scientist. The third one, which is the fourth one, which is promising, is now being manufactured by Serum Institute. Now, this is an Oxford University vaccine, and AstraZeneca is the one of the companies who are the main company with the Oxford University. This vaccine has got a platform which is the shimp adenovirus platform, because there is a problem with using adenoviruses. We have a lot of antibody in our population. So normally you look for adenoviruses which are not there in the population. So this shimp adeno platform is being used by the Oxford. They are very excited about their vaccine, and they have given to Serum Institute to manufacture it. Although some people, they have given to 5, 6 other companies to manufacture it also. Serum Institute made an announcement, and Adar Punewala went into the TV to say that they will be making it, and they have started making it. They promised that they will manufacture it within this month. There will be manufacturing batches which will be coming out of that particular place. So this is a vaccine which will be available, and this vaccine is also will move. We do not know the nature of the agreement over here. There are some kind of a speculation which was going on in the buzz world, but I will believe when Mr. Punewala says something, he stays with the full responsibility, and this vaccine will be available in India and other parts of the world very quickly. This has also moved into phase 1, and at the moment the phase 1 data has not been posted. So mind you, none of the phase 1 data has been posted, not even the Moderna phase 1 data. They're going to post it within few days. The fifth vaccine, which is of very good progress, is the Johnson and Johnson's vaccine. Johnson and Johnson's platform is adeno 26. This is very rare adenovirus, is not present in the population. It doesn't have oncogenicity, and they are using the prefusion spike protein in their vaccine. So this vaccine has undergone, they are very hopeful that this will move very fast. So the Johnson and Johnson has brought out the Ebola vaccine. That Ebola vaccine has been used in the population. So they are very confident that they will be able to use it. Their RSV vaccine is also in the phase 3. So that is also in this particular platform, and they're confident that this particular vaccine will also move appropriately fast. They have not gone to any other partnership, but one of the impending partnership which is happening is between Aurobindo and CSIR Imtech, that the prefusion protein which will be manufactured there of the spike protein will go with an adjuvant. So this will be an antigen which will be going with a strong adjuvant for the immunization. That also is one of the vaccine strategies, but it is in very early stages. But Aurobindo has acquired a small company in USA which was a hive-off from the Pfizer, and they have a vesicular stomatitis virus platform. It is a very potent and beautiful platform. So Aurobindo in Hyderabad, which is new in the vaccine area, has got a huge vaccine company which has already been made in Hyderabad, and are moving towards the carbohydrate vaccines, like carbohydrate conjugate vaccines by like pneumococcus and other vaccine. They want to enter this. They also want to enter into the HPV arena, but they also want to enter here, and they are in a considerable progress. They are the preconcept or the concept vaccine is already available with them. Then let us move to our other company known as Bharat Biotech. Bharat Biotech recently has been funded by the CSR Nimitli. Bharat Biotech has partnered with CCMB in an inactivated virus vaccine. It is the easiest thing to produce. They will be doing it in the virocell. They have a lot of experience with the virocells, and this particular experience will be very useful because they have the certified cell lines. It takes a lot of time for certification. They have used it with the rotavirus. So they have confidence that they can enhance it. They have also a cryopreservation technique which will make this antigen stable in the for the use, which is very important part in the vaccine preparation and generation. So this particular thing will be moving very fast because the CCMB is a good vaccine manufacturer, particularly the novel vaccine manufacturers in India. And the partner here, the CCMB, is very strong. Zydus Cadila also has two different partnerships at the moment, and they are moving. One also, there is another molecule with the Serum Institute, which will be an attenuated variety. But one of the important platform of the Zydus Cadila I will like to mention, they are using the measles virus platform for the preparation of this vaccine. Now, this creates a really very good system. If you have a measles virus platform, then you can give it in a EPI mode along with the measles virus. And if you go for a 3 successive years, you will actually immunize the entire population, because birth cohorts in India are 27 million. So if you do 3 or 4 rounds of 27 million each, you almost cover the generations of Indians with this vaccine. We do not know how long the vaccine antibodies will last. The phase 1 will be followed up to 1 year, and the same way the phase 2 will also be followed up. So we do not have the data for that. Normally for respiratory viruses, you need another booster and immunization in this every second year. Many viral vaccines, immunization is for life, like smallpox vaccine, measles vaccine, mumps vaccine. So many vaccine, it is for the life. And here we do not know that how long will it last. And hopefully, if it lasts longer, it will be good for us. If it doesn't last longer, then we do not know. But we should, at this given moment, prepare that we will need it every second year. But one of the most important group which will receive this vaccine as soon as the vaccine comes up are the first responders. They will be the nurses, ward boys, doctors, those who have come in contact, as well as the other ancillary staff who go as a first responder, the cleaning staff, the sanitary staff, and other staff, because that makes a sense. And the dentists as well as the anesthetists, who are the most vulnerable population amongst the doctors. So they are going to receive it the first. Then there could be other groups. We might like to protect groups which have the underlying comorbidities, old people with comorbidities. The problem in our country is this, that old people, like people of my age, don't exist officially. We are just unofficially existing in our country. So at this given moment, we need to have a robust vaccination program for this. We need to also prepare for vaccination, because during the November and other, the influenza as well as this particular COVID infection is going to be together. This infection is not going away anytime. We have to learn to live with it, with appropriate PPEs, appropriate self-protection, appropriate behavioral insights, and appropriate quick kind of intervention wherever we need, and course corrections will have to be done. The maps will be redrawn. So we will live with it. But at this given time, this should be this could be the sequence of eligibility of the vaccines.
Sir, I want to intervene. This is too advanced for us. So we would like to have one more session with you next Saturday.
Okay, so I will round up now.
No, sir. You will round up. I'm saying is that this is something which we will take at least 2 sessions to understand you, not one. So we'll request you to one more session on next Saturday at the same time. We will read about it in great detail because we will like to ask questions 1 full hour because I'm meeting all the notes, and this is something which I need to communicate in a single language with yours. I will write a complete review with you so that all the doctors in the country can understand. So I'm only requesting you to sum up, and then we will have another session next Saturday, same time.
Okay.
Here we will ask the question because it is, we really need to go because we have never been taught the way you are teaching. Thank you, sir.
Okay, so I will wrap up with this, that other vaccines effort could be that we make it a disease modifying vaccine. Those who are infected already, we give this vaccine so they receive antibodies and we could save them. So this is another mode of vaccination. At the moment, we don't have any trial on that, but this is in the drawing board. The second thing in the best to the vaccine are the immunoglobulins. The Israel, as you know, has created an immunoglobulin which is widely reacting to COVID-19. At the moment, the mutations are not enough so that we could be afraid of that. But we should be ready that, like influenza vaccine, if major mutations occur, we could do the course correction without much regulatory activity. So monoclonal antibodies, which are being made by many companies around the world, 2 Indian companies are also making it, and several Indian institutes like National Institute of Immunology, etc., also engaged in this. So this is another method of moving. The CAR NK cell is another major area. There are few major effort on that, and National Institute of Immunology in India is also moving with the CAR NK cell. So this is something which is also we need to watch appropriately. Finally, I will say 2 things here. That one of the things are the immunomodulators. So BCG is moving around in many countries. Stephen Kaufman's vaccine, which is made by Serum Institute, which is again a mycobacterial vaccine, is moving. But one of the major good vaccine, which is known as MW vaccine, which is made by Cadila, was developed by NII, has been the first successful vaccine for septicemia. There's no other drug which has acted on septicemia. So this has undergone 3 trial things. One is a prophylaxis, one is for moderate, and another for critical patients. This immunomodulator we are seeing with a very close eye. But PGI, which did this vaccine trial, has already treated 15 patients. So I'm finishing it now. That two patients, they have published in the Lung India, and 14 patients who have become they have recovered will be published very soon. So this is at the moment the overall review of the various kind of efforts which are going around the world. Many of them are also being followed by Indian companies, and there are 3 or 4 different strategies which are moving up. Thank you for this opportunity. Thank you very much.
Thank you, sir. And it was a wonderful talk for people like us. I'm reading about it for the last 4 months. I learned a lot. We'll read about it for a week, and next Saturday we will be interacting with you on this in great detail. I'll request Dr. Ashokan to propose a vote of thanks to Dr. Ganguly.
Yeah, it was really a...
Dr. Agarwal. Dr. Agarwal. Hello.
Yes, Arun, we will have to discuss.
We have the...
Yeah. No, I have invited my scientists to just brief about how the software...
Sir, I think we will have it in our next section. Right, Royally, as a guest.
So, Dr. Agarwal, I have a small request. Hello,
Hello.
Yeah.
Hello.
Dr. Agarwal and Dr. Ashokan, I have a small request
Yes, sir.
that if you have recorded it, if you could really make this available to me, the link is make available to me so that I could go through what I said, because I went on talking extempore without preparing, and I want to really see whether everything I said is correct or some of the things need to be
That will be done.
corrected in the
Before we
future.
put it up in the website, we will do
Okay.
that.
Okay.
Dr. Arun, you want to make a point?
No, no. I asked my scientist to just to brief. If you unmute her, she is Madhura Tatode. Just she will brief about 2-3 minutes about how the software technology platforms can be useful for any vaccine manufacturer. Just unmute her.
Sanjay, you are on the line. Sanjay, you are on the line.
Hello.
Madhura, go ahead.
Go ahead.
Just
Go
brief,
ahead.
because our platform is useful for all vaccine manufacturers, and through this platform we will be able to reach to everybody. Just brief about what we are doing, keeping in mind NDA.
Yeah, sure. So this is Madhura from Persistent Systems Pune, and basically I'm bioinformatician. So as a bioinformatician, considering it's very trending and it has increased the scope because of easily available genome sequence data, we are trying to develop one platform which will be end-to-end analysis platform before going ahead for the in vitro synthesis. So whatever we are trying to do is before whatever we are doing at the time of in vitro synthesis of vaccines. And here what we are trying to do is considering genome sequence data, because it is easily available nowadays and China has already released the reference sequence of that. Considering different bioinformatics resources, we are gathering the genome sequence data from that and trying to predict the epitope sequences. Considering epitope is very much important step before going ahead for the vaccine synthesis. So our platform will take into consideration epitope sequence, and in the output it will give one final analysis report where we will have all the detailed analysis by consolidating different bioinformatics analysis algorithms. So this is what our platform is. But it will be really very much helpful if we have this brief idea to discuss, considering a young or young bioinformatician which maybe contribute to the COVID-19 analysis. So basically, why we were trying to contribute to this is virus has very much high rate of, and it is very much important to have particular epitope sequences targeting towards the viral variations. So, considering this need and its continuous global spread, it is very much important to reduce the time of vaccine development. So, considering this need, we have proposed the idea to expedite the process of vaccine development using different bioinformatics algorithms. So this is where our platform comes into the picture, and this will be really helpful not only considering COVID-19 but also in terms of different viral particles.
Madhura, will it be useful for DNA vaccine?
Exactly. So this will be basically useful for DNA vaccine, and since this is bioinformatics approach, it will use sequence data as an input.
Yeah, so you can use for any type of vaccine.
Yes, we can use it for. So this is basically US technology approach which we are using, and we are considering known experimental data from IEDB bioinformatics resource where they have known epitopes we shown to have immunogenic response across different animal models. So we are considering homology based approach here along with prediction of novel epitope candidates. So if we have any homolog for the, let's say in case of COVID-19, considering spike protein which is actually responsible for its entry into the host cell, homolog of that SARS-2 coronavirus spike protein is spike protein of SARS coronavirus, which was there in 2002 endemic. So considering this, it will be very much useful to understand and to check the cross reactivity, or if there are any epitope candidates which might be called it as potential vaccine candidates, which will be useful in case of COVID-19 case also. So this is one approach, and second was to predict the novel vaccine candidates, again using different bioinformatics analysis pipeline. So using this different bioinformatics analysis pipeline, it tried to predict the epitope sequences from input protein. So we did small study for spike protein, where we were able to fetch final 6 to 7 epitope candidates based on different amino acid physiochemical properties, along with different artificial intelligence based algorithms. So this is platform to expedite the process of vaccine development, because we all know that vaccine synthesis till clinical trials take so much of time, and it is very much important to reduce the time for that. So bioinformatics is the best suited thing for this, and this is why, because it is very trending and genome data is very easily available in a current days. So yeah, that's it. So this is the screening platform, epitope screening platform.
So,
I think we should conclude now. I'm grateful for the intervention of Dr. Arun, and I'm grateful to Dr. N.K. Ganguly. We close right now. And as Dr. K.K. Agarwal said, we will have another session exclusively for questions. Before that, I will send a recorded copy to Dr. N.K. Ganguly for his vetting and approval. Thank you so much. Thanks for everybody for joining us on this webinar. Thank you so much.
Thank you for opportunity. Thank you very much.